Despite years of intensive therapeutic efforts for patients with muscle-invasive urothelial bladder cancer (MIBC), 50–60% of the patients with basal and luminal subtypes will develop local or distant tumor recurrence within 5 years. Therefore, it is essential to further elucidate the underlying mechanisms of tumor development and determine which signaling gene pathways are important during the process of tumorigenesis to identify new therapeutic targets. In this study, whole organ bladder mapping was done on 36 separate tissues excised from a whole bladder involving an entire excised bladder housing urothelium, precursor lesions, and different stages of carcinomas were globally mapped both histologically and molecularly using mRNA and protein analyses. In addition, a separate cohort of patient-matched tissues comparing precursor lesions, mainly carcinoma in-situ (CIS), with matching muscle-invasive tumors was analyzed. As a result, this study concluded that the final stage of subtype commitment to a luminal or basal MIBC occurs at the transition from carcinoma in-situ to invasive tumor stages, which is a complex transdifferentiation process where CIS frequently showed preserved expression of both luminal and basal differentiation markers. However, marker expression of the final subtype occurring in MIBC was dominant in basal or luminal CIS, respectively. Moreover, aberrant staining patterns of luminal and basal immunohistochemistry markers are helpful to distinguish between luminal or basal CIS. In addition to luminal and basal gene expression phenotypes, this study brings forth novel findings that precursor lesions and tumor-associated urothelium share a similar gene expression, defined as a luminal extracellular matrix ECM-P53-like gene expression phenotype. Normal urothelium from healthy non-cancer patients had significantly lower ECM-like gene expression, supporting an oncogenic field affection of normal-appearing normal urothelium, and a significant impact of the stromal tumor environment in tumor development and progression. Investigating the luminal ECM-like expression phenotype further using a pan-cancer progression panel of 681 genes revealed pathways specific for ECM remodeling, angiogenesis, epithelial to mesenchymal transition (EMT), cellular discohesion, cell motility, supporting a strong role of these pathways in tumor progression already at the stage of normal-appearing tumor-associated urothelium. On the other hand, genes defining cell proliferation and oncogenic ERBB2, ERBB3 signaling were prominently upregulated in invasive bladder carcinomas. This study is important as it paves the way for clinical translation with the hope and possibility of identifying genes that can be targeted for MIBC therapy, especially for challenging clinical cases with high rates of recurrence and progression.